As you see long roots extending from the onion. Now, if you have read about roots, how the extension takes place, it's just mitosis. It's just, cells are dividing rapidly, and most rapidly dividing cells you will find here, which is the point of growth, which is the point of extension of root. And you can take these root tips and then you can squash them and then visualize under the microscope. You need, as I said, you need dye. We use acetylcarmine. We use dye to stain cells. We need microscopic slide. Glass slide. We need cover slips, and we definitely need a microscope, a simple compound microscope, not a very high-end microscope. There are different kinds of microscopes. When you come to our department, you will see compound microscope, then more advanced kinds of microscopes, like we have the confocal microscope, where we can go within the cell on a single point, for example a dot on a chromosome, DNA binding sites of certain genes. But for visualizing different stages of mitosis, we use a simple compound microscope, not a very specialized one. Then what we do, we take this root tip. This root tip is actually, let's write down the next thing in methods. So this was the methods part, when you took onion, put it in beaker of water, held it in such a way that the onion does not drop directly into water. Its roots, the bottom part just touches the water, and then it will start extending, the roots will start emerging. This experimental setup, so you have the grow onion roots. Then with a fine scalpel or a blade, you just take tip of the onion and eventually you squash it. Squash onion root tip, place it under the cover slip, and then with a thumb you can very easily smash it. It's very succulent, it's very tender, and all the cells which will be growing here in the onion root tip, which are rapidly dividing, there you will find different stages of mitosis. You need to put a dye which is going to stain all the chromosomes, and after staining with this dye, once you will undergo a microscope, you will see all these different stages which we have here. For example, you will find cells in metaphase stage, anaphase, telophase, all different stages of mitosis you will be able to visualize. At your level in BIO100, we don't study meiosis. It requires more, time, because but once you visualize the stages of mitosis, stages of meiosis are more or less the same. But that can be also easily done if you wish. You have to use what kind of cells to study meiosis? Can you study meiosis in the onion root tip? Let me take a pause of few seconds to let you all think about if you can study meiosis, reductional division, and then the meiosis 2 and then 4 product of meiosis, like generation of haploid cells from diploid cell. Can we study using onion root tip? So if not, the question is why? And if we will not study meiosis in onion root tips, which tissue we will use to study meiosis? So I leave you with that question. I'll not answer. You should answer these questions to your lab instructors. Let me wrap this lecture up by stating that whenever you design an experiment or think of performing an experiment, you need to spend maximum time on the design part. You need to think about a very clear objective. You need to think of, what kind of tools you need, materials you need, how you will perform the method, how you will perform. Do you have the necessary skills to perform that experiment? If you are not skilled, how you will learn them? So at your stage, we are just trying to fascinate you about different biological experiments we perform or we use in cell and molecular biology, or in next few modules you will learn about genetic engineering. Genetic engineering is just a very tiny part of what we do in cell and molecular biology when we try to understand human development, fly development, C. elegans development. We try to understand diabetes, we try to understand cancer, we try to understand how we develop as a normal human. Okay, so all these things will come across as we move on in this lab. But whenever you think of being a student of science or going to do an experiment on the bench, whether it's a lab or as a grad student, you have to be very critical. Because once you will see the results, after methods, you perform experiment. After performing experiments, you have to take data. You have to, collect all the data very honestly. You may be thinking of, a hypothesis, but your results are turning out to be totally opposite of your hypothesis. So science is all about finding truth. Science is all about, finding how nature works, how a living system works, how a biological system works. It may work against our hypothesis. So once you get results, then the analysis part starts. So your lab notebooks, they should have, a clear objective or the experiment, then the materials you need, the methods you are going to use, the protocol, the experimental protocol, which is basically the method how you are going to perform experiments, then eventually the results you get out of that, and finally conclusion and analysis of your results. So that's how you should maintain your lab notebook, and that's how science is actually done, whether you're in biology or you're in any scientific discipline. So thank you very much, and I hope you enjoyed this session. I ask you all to stay connected with your lab instructors.